Namaskar to all. In our previous video we learned how to draw this plant cell, and now we will learn how to draw an animal cell. So in the same way we will draw animal cell. First thing, we will remember all the details which are necessary for making an animal cell, and next thing we draw only those details which we want to show and what is our purpose. So if we are making diagram for exam, so what is the need of the question. So a simplified diagram we can make. First thing, that plant cell is having a cell wall. It have some rigid structure, rigid cell wall. Animal cells do not have any cell wall, so you can draw any flexible structure which may be spherical, may not be spherical. So make any random structure. This is an animal cell. So there are variety of animal cells, so you can draw any structure. Next thing, because plant cell is having cell wall and animal cell do not have any cell wall, it have only cell membrane. So you can, this simple line will serve as cell membrane. So this is our cell membrane. Now we have the most important part of any cell, which is nucleus, which have a membrane with pores. So we draw discontinuous line. So these discontinuities show pores or holes on the nuclear membrane. And one thing we need to remember, that all things are spherical. So plant cell or animal cell is spherical. So if you see cell membrane from outside, you cannot see an inside material. And if you are able to see nucleus, so you cannot see the material inside nucleus. You can only see these pores like this. But we are not making here three-dimensional diagrams. Sometimes we see three-dimensional diagrams in many books, so for clarity in the concept. So here we are making two-dimensional structure, and we are using only symbols to convey our message. Next thing, so these continuities are nuclear pore, and we know that nucleus is having double membrane. So we will draw like this. And we have rough endoplasmic reticulum and smooth endoplasmic reticulum in an animal cell, which is having continuity with the nuclear membrane. Sometimes it have, sometimes it do not have. So we will make some continuity, because when we see nuclear membrane or endoplasmic reticulum in our textbooks, sometimes we see smooth endoplasmic reticulum or rough endoplasmic reticulum attached to it, sometimes we don't. So here I will, because I am making 4 parts or 4 sections in this membrane, so I will attach 2 sections to show these structures. So here are our endoplasmic reticulum. So we will add ribosomes to show whether it is rough endoplasmic reticulum or smooth endoplasmic. So this is my rough endoplasmic reticulum. In the same way I made ribosomes in plant cell, I will make ribosomes in animal cell also. So I will make these ribosomes little bead like. Why? Because if I want to show cytoplasm, so I will make like these small dots, so we can differentiate whether it is cytoplasm or ribosomes. Now we have most common and important and easy to make structure, which is called mitochondria, and we have many mitochondria in our cells. So I will make 2 or 3 mitochondria, sorry, not mitochondrion. Singular term is mitochondrion and plural term is mitochondria. So I am making mitochondria. 3 are enough. Now okay, one more thing, we can make vacuole in plant cell also, we can make cell vacuole in animal cell also, we can make lysosomes in animal cell also, we can make lysosomes in plant cells also. But what I will do, I will not draw any lysosome in plant cell, and I say in the same way I will not draw any vacuole in animal cells, to make us clear that okay, it is not important to have lysosomes in plant cell, and it is not important to have vacuole in animal cell. So I will make some spheres, and I will label these spheres like lysosomes afterwards. Next thing, if inside nucleus, same thing I will make. So in plant cell I made this nucleolus. We can label this also if it is required. If we are in higher classes, in class 9th or 11th, then we can label it. otherwise there is no need to label it. And then we can make a rough meshwork, and this is our chromatin inside nucleus. So now it is enough for nucleus. Now we have more endoplasmic reticulum which is not attached to nuclear membrane. So here is our endoplasmic reticulum, and this is rough endoplasmic reticulum having ribosomes attached on its surface, and this is why it is rough endoplasmic reticulum. Okay, and it is smooth endoplasmic reticulum. Now we have enough structures to label. So this is very simple to make, and we are having no chloroplast. We are having chloroplast only in plant cell. So this is so simple to make any cell for examination purpose. We can add more things to fill it. We need Golgi body. So we have not drawn yet. We can make. I will draw here so that you can see how I make it. So it is like endoplasmic reticulum, but it have a specific symbol when we use in our books. This is a Golgi body. We can make, because we have space left here, so we can add more Golgi bodies. We can make more mitochondria also, because it is feeling little empty. And we have more lysosomes, more mitochondria, and more endoplasmic. Or we can make it clear also, because it is if the diagram is clear and we label it properly, it will look good. So I will label this by following the same pattern. I will label in right side. Okay, so here is the outermost layer, which is called cell membrane or plasma membrane. Next we are having this mitochondrion, or mitochondria we can label. These are lysosomes, and if you want you can make cell vacuole also, but if you are making cell vacuole, then it must be small, not this large. So this is the difference between plant cell and animal cell, having large central vacuole. Okay, next this is nuclear. You can write nuclear membrane also, you can write nucleus also, it depends on you. So if you want to label only nucleus, then you can write nucleus. Otherwise, if you are in class 11th, you can label more. This is a nuclear membrane. This is nucleolus. So you can label according to the need. This is smooth endoplasmic reticulum, and this is rough endoplasmic reticulum. Okay, now what left? Golgi body. Okay, so we can make more. Either we can label from here, or we can make one more Golgi body to solve this issue. So here we are having. So I'm showing you that okay, it is very random. There is no fixed rules how to make cells, but there is no need to make as it is from textbook. But these are only symbols. So examiner want to know about your knowledge of the cell. So it is Golgi body, or Golgi apparatus. Okay. So this is our animal cell. I am sorry. So here you can write the name of the cell. That this figure is animal cell. And if you want to show the difference between prokaryotic cell and eukaryotic cell, you can also write it is a eukaryotic cell, or you can write eukaryotic plant cell. Okay, This is also a eukaryotic cell. So this is how we draw an animal cell, and by drawing these diagrams you can also notice the differences between plant cell and animal cell, which are very important also. You can make a table of this also. So plant cell is having cell wall, animal cell do not have any cell wall. Plant cell have large central vacuole. Either animal cells do not have any vacuole, or you can make a sphere and you can label it as a vacuole. Or if they have a small vacuole. Okay, let me label it also. Let's label it. Okay, there may be vacuole, but it is not as big as this. Okay. And we are having lysosomes in animal cell. We are not having lysosomes in animal cell. If we are having, because this vacuole is doing that work. And chloroplast, definitely the cell organ which makes plant cells green, or because of that plant cells are able to photosynthesize. So topic is not the difference, but you can know the differences. So while drawing these diagrams you can cover many topics, but they all topics are interrelated. So thank you so much for watching. I hope you learned something, and you can see while drawing diagrams you can label more if you want. So you can label like nucleolus, like chromatin, nuclear membrane. So you can make label anything. Ribosomes, I think I have not labeled ribosomes on the surface of rough endoplasmic reticulum, and you can make ribosomes spread in cytoplasm also. So here are our other ribosomes. You can label it as ribosomes. Very important. Okay, so I will end this video here. I hope you enjoyed this video.